The experimental proof of semi-conservative method of DNA replication. First carried out by Meselson and Stahl. Meselson and Stahl in Escherichia coli. First experimental proof. DNA replicates semi-conservatively, carried out by Meselson and Stahl in E. coli, Escherichia coli. Experimental material, Escherichia coli. Chemicals used by them: cesium chloride, N15 contain ammonium chloride, N14 contain ammonium chloride. Chemicals used by them: cesium chloride, N15 contain ammonium chloride, N14 contain ammonium chloride. Then what is the use of cesium chloride? Cesium chloride is heavy salt. When dissolved in water and when spun at high revolution, that is 50,000. revolutions per minute, it produces high concentrated part. It produces high concentrated. Any other substance which present above that one only. That means it is used to observe the DNA clearly in the test tube. Always cesium chloride present at the base in the test tube and the DNA that present always above that cesium chloride level. Because of the high concentration or high density of this cesium chloride always present at the bottom of the test tube and remaining any material always they are less density present above that one or toward the surface. That is the advantage of utilization of the cesium chloride. Now, first the bacteria cultured in N15 contain ammonium chloride. So, all bacteria are incorporated with N15. N15 that incorporated in the DNA and other nitrogen containing substances. And then this bacteria that taken into the test tube, cesium chloride, and centrifuge. So, DNA that separates, it consists of only N15. So, that is heavy DNA. The DNA with only N15 called heavy DNA. Next, this N15, next the bacterium cultured on N14 medium, N14 nutrient medium for many generations. Then, after 20 minutes, then they centrifuge, it is the N15 plus N14. Hybrid are obtained. Generally, bacteria replicate at every 20 minutes. So, obtain N15 plus N14. N15 plus N14. That is the hybrid DNA. DNA that contains both N15 and N14. That is the hybrid DNA. Again, another 20 minutes. Suppose it is the first generation, it is the second generation. When the bacteria was centrifuged, It is hybrid and it is a light DNA. The hybrid and light DNA. Now we analyze this one. N15, both strands are N15 contain. In the first generation, it is N15. New strand that synthesize consists N14. It is the first generation. In the second generation, N14, 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 N14. So, two hybrid DNAs and two light DNAs are formed in the second generation. Next generation, that is the second generation, two hybrid DNAs. That means number of DNA molecules that form in second generation 4. In third generation, double 8. Out of 8, 2 are hybrid, 6 are light. So the thickness of light increases year by year, but the thickness of hybrid is always constant in any generation. Thickness of light DNA increases generation to generation, but the thickness of hybrid is always constant in any generation. In the parental generation, no hybrid, no light DNA. In the first generation, only hybrid DNA without light DNA. From second generation onwards, we observe both hybrid DNA and light DNA. Both hybrid DNA and light DNA. Now, what is the percentage of hybrid DNA and light DNA in various generations? Now, again listen here. Chemicals used by Meselson and Stahl. N15 contain ammonium chloride and N14 contain ammonium chloride. First they grew in N15 contain ammonium chloride. So DNA N15 incorporated the DNA, and then he centrifused, he obtained heavy DNA. Allowed to the DNA into the N14 contain nutrient medium. After 20 minutes, when the bacteria are centrifused, obtain hybrid DNA only. After 20 minutes, in the same N14 contain ammonium chloride medium, the bacteria grow and then centrifuge, obtain both hybrid DNA and light DNA. In the next generation, that is the third generation, the thickness of light DNA increases, but the thickness of hybrid DNA is always constant. Now we observe the analysis and the percentage of this hybrid DNA and the light DNA in different generations. Both strands consider N15 in parental generation. First generation, the two strands separated, N15, new strand synthesizes, N14, first generation. Second generation, N15, N15. N14. Now, the percentage of hybrid DNA in parental generation 0%, no hybrid DNA. In first generation, both strands are hybrid, 100%. In second generation, out of 4 DNA, 2 hybrid, 2 light. There is a 50%. It is the parental generation, first generation, second generation. If we continue this one, 25% hybrid DNA in third generation, 12.25 in fourth generation. That means generation to generation, the percentage of hybrid DNA decreases into half. The percentage of hybrid DNA decreases into half generation to generation. And the ratio between hybrid DNA and light DNA. Ratio between hybrid and light DNA in parental generation. There is no hybrid DNA and light DNA. So that is the 0:0. In case of the first generation, only hybrid DNA present, no light DNA, that is 1:2:0. It is the hybrid and it is the light. In case of the second generation, 4 out of 4, 2 hybrid, 2 light. That is 1:1 ratio. In second generation, 8 are formed, 8 DNA molecule. Out of 8, 2 are hybrid and 2 are hybrid and 6 are light. So ratio is 1:3. In the fourth generation, 16 molecules are formed. Out of 16, 2 are hybrid, 14 are light. So ratio is 1:7. So ratio between hybrid and light DNAs in parental generation 0. Ratio between hybrid and light DNAs in first generation 2:0. Ratio between hybrid and light DNAs in second generation 1:1. And the ratio between hybrid and light DNAs in case of second generation, third generation 1:3. And the ratio between the hybrid DNA and light DNA in fourth generation 1:7. That is the number of DNA molecules formed in n number of generation, that is 2 to the power of n. N is equal to number of generation. For example, in the fourth generation, that is the 2 to the power of 4, that is 16. Out of 16, in any generation, hybrid DNAs are always 2. Remaining DNAs are light DNAs. That's the reason the thickness of light DNA in Meselson and Stahl experiment, thickness generation to generation, but the thickness of hybrid DNA is always same in any generation. So Meselson and Stahl conducted this experiment first in E. coli, and then this conservation of semi-conservative method of the DNA observed in remaining all organisms, even in eukaryotes also. Scientists who first observed the semi-conservative method of the DNA replication in eukaryotic cells by Taylor